For the next presentation is the roles and underlying mechanisms of homologous recombinant repair protein RAD54L in mediating tumor immune escape by inhibiting IFN-I signaling by Xiaoke Wang from Naval Medical University.
Respected judges and friends, my name is Wang Xiaoke, grade 4th from the Naval University, and today my topic is homologous recombinant repair protein RAD54L mediates tumor immune escape by inhibiting the type 1 interferon signaling pathway. I will present my report in the following 4 parts. Let's begin with the background. In the struggle between the tumor tissues and the immune system, the crucial epigenetic alteration is the call for tumor to achieve immune escape. To explore the crucial epigenetic alteration that play a key role in the tumor immune escape, we explore the kidney cancer patient cohorts in the public databases for the epigenetic related genes that are differentially expressed in the normal tissues and the tumor tissues, and whose expression level is highly related to the patient's prognosis. Then we notice the molecular RAD54L. In the previous studies, RAD54L expression level is highly related with the occurrence and development of a variety of tumors, but whether it plays a key role in the tumor immunity has been poorly understood. On this basis, we conducted the following design, including the bioinformatic analysis, the in vitro and in vivo experiments. And here are our experiments and results. Firstly, through the bioinformatic analysis, we confirmed that the RAD54L was highly expressed in a variety of tumor tissues. And further, we verified this in the patient's tissue samples. From the kidney clear cell carcinoma through the immunohistochemistry. So we got our first conclusion. RAD54L was highly expressed in the kidney renal clear cell carcinoma and indicated poor prognosis. Subsequently, we generated the knockout cells for the tumor-bearing experiment. Interestingly, the RAD54L knockout suppressed the tumor size in the biopsy mice instead of in the nude mass. Furthermore, the knockdown of RAD54L increased the CD8 active T cells infiltration and activation in the tumor microenvironment. Thus, we got our second conclusion: loss of RAD54L inhibited tumor immune escape. Subsequently, we conducted the mechanism investigation, and through the RNA sequencing, the RAD54L knockout regulate the type 1 interferon signaling pathway. And further studies show that the knockout of RAD54L increased the level of the ARF3 activation and interferon-related genes. And through the flow cytometry and immunohistochemistry, we found that the RAD54L knockout increased the level of cytosolic dsDNA. And moreover, the C-gas knockdown can significantly inhibit the promoting effect of RAD54L knockout on the type 1 interferon signaling pathway. This result suggested that RAD54L knockout induced cytosolic dsDNA accumulation and activated the type 1 interferon signaling pathway. Finally, we through the prediction and analysis of the transcription factors of the RAD54L, we found that the FOXM1 regulated the expression level of RAD54L, and the inhibitor of FOXM1, we called it thyrstreptin, can downregulate the expression level of RAD54L and can release its inhibition in the type 1 interferon signaling. And more importantly, targeting FOXM1 and RAD54L can inhibit the tumor progress in the tumor bearing mass and can increase its survival rate. This results suggested that the inhibition of RAD54L expression was a potential treatment strategy for tumors. In summary, by upregulating the expression level of RAD54L, the tumor cells can reduce the cytosolic dsDNA levels, thus inhibit the type 1 interference signaling pathway response to achieve immune escape. And the loss of RAD54L and the inhibition of it can increase the level of cytosolic dsDNA accumulation and thus activate the type 1 interference signaling pathway to trigger anti-tumor immunity. Our study also offers some innovations. For example, our study deepened understanding of the relationship between the homologous recombinant repair and the tumor immune escape, and also provide a theoretical basis of the tumor treatment strategy which targets the RAD54L. And in the future, we plan to verify the effect of the RAD54L in other types of tumors and investigate the therapeutic potential of combining serostreptin with the PD-1 antibody. That's all for my presentation. Thank you for your listening.
Okay, thank you very much for your excellent presentation. In your approach here, the RAD54L, right, suppressions approach. Can you discuss the risk of this approach? Because here you try to enhance the genotoxicity, right, to increase the production of DNA and activate immunoresponse. But here, in the same approach, it also can introduce the genetics escape from the cancer cell as well, right? How you balance these two arms, whether it's going to be better killers or is help the cancer to evolve and escape in another way.
Yeah, we also consider about this problem because in the previous study, some studies show that in the activation of the type 1 interference signaling pathway, which caused by the DNA damage or the DNA the repair inhibition by some therapies such as the chemotherapies, the radiotherapies, it can also lead to these effects. This effect can increase the expression level of the PD-L1 in the tumor size to let it to achieve the tumor immune escape. So we studied some articles previously. So we mentioned about we want to try to combine this with the PD-1 antibodies to avoid its side effect, we call it, and to get it more practical to the therapeutic treatment. Yeah, thank you. Any questions? Okay, very thank you for your listening.